So I am a fourth-year PhD student in the Ocean Science and Engineering new interdisciplinary PhD program. And my talk will be on deep subsurface bacterial proteins that bind and modify clathrates. Next. Thanks, Jen. So methane clathrates are basically cages of water molecules that trap a single guest gas molecule. So in the case of methane clathrates, that would be a methane molecule. These occur at high pressure, low temperature, and wherever there is abundant gas. So on Earth, that's along continental margins and permafrost. And they're also predicted to be on other planetary bodies like Mars. So this is a cross-section of Mars and the potential methane sources and sinks. And you can see in the middle there where the subsurface is, they predict a methane clathrate reservoir that could be getting its methane from microbes producing methane or a water rock reaction called serpentinization, which would then outgas into the atmosphere, where it's then pretty quickly degraded. On Earth, we have a problem in which gas clathrates actually form in natural gas pipelines, and they'll actually clog and explode the pipelines. This is really dangerous, and it was actually the cause for the Deepwater Horizon oil spill because the clathrate clogged the wellhead. So yeah, definitely lethal and a serious environmental hazard. One of the solutions has been to use gas clathrate inhibitors, and commercially they use things like methanol and PVP, but now they're looking for a more green or environmentally friendly clathrate inhibitor, and they found that antifreeze proteins, which were evolved to bind regular water ice and organisms, actually bind gas clathrates too. So one example is the type 1 AFP, and it's this alpha helical protein that's secreted in the blood of the winter-flounder, and it binds to nucleating ice crystals, and it keeps those crystals from growing and freezing the fish in sub-zero water. Another example is the type 3 AFP, and you can see the structure is totally different, even though it's in an eel pout, probably evolutionarily close. And then another example is this beta-solenoid protein, which is found in a beetle. Next. And they found that it's the type 1 AFPs, those alpha helical AFPs, inhibit gas clathrates most effectively. And they do this by using this amino acid motif, this threonine, alanine, alanine motif that actually get inserted into these empty cages of the clathrate. So we know that there are microbial consortiums in methane clathrates, and we also know that ice-dwelling organisms evolve proteins to bind ice, so maybe gas clathrate-dwelling organisms evolve proteins to bind gas clathrates. Next. So we hypothesize that there are bacterial proteins from methane clathrate-bearing sediment metagenomes that are optimized for binding to gas clathrate, and we could call them clathrate-binding proteins. Next. So we have sequenced microbial DNA from clathrate-bearing sediment cores from Oregon, and off the coast of Japan as well, from the Shimokido Peninsula. And from that metagenome, which is the DNA from the entire microbial community, type 1 antifreeze proteins were actually top hits to several of those bacterial proteins. And so we chose 8 of those proteins to express in the lab, in Raquel Lieberman's lab. Justin Heward did this, and they were expressed with green fluorescent protein in E. coli. And we expressed with green fluorescent proteins so that when we did experiments in the lab, we could actually visualize the protein binding to the clathrate. Next. So for part 1 of my thesis, I tested the purified clathrate binding protein, or CBP, activity on this type of clathrate called tetrahydrofuran, or THF-clathrate. It actually doesn't occur in nature, but it's easy to work with. You don't need a pressure cell. It's liquid. So we machined this drainage-capable beaker where we actually injected a THF solution and nucleated a THF-clathrate crystal at the end of that pipette using dry ice. And so we used different treatments. We used negative controls, including eGFP by itself, that green fluorescent protein, as a negative control. We also used type 1 AFP as a positive control. And we tested several of the CBPs, or clathrate binding proteins, putative at this point. So this is what our negative control looked like. This is THF-clathrate grown in the presence of that green fluorescent protein. It looks like no additives were included. It is regular, octahedral, beautiful, and we even shined blue light on the crystal. That's what you see in that corner there. And we saw no fluorescence, indicating that the GFP did not bind to the surface of the clathrate. As our positive control, we grew clathrate in the presence of that type 1 AFP, and we see a very different morphology. It's more like platey. It's going in like each crystal is going in one direction. We think that might be because the protein is binding to one or two planes of the clathrate, allowing it to only grow in one direction. So then we tested our CBPs, and CBP3 bound to the clathrate similarly to the type 1 AFP. So on the left is the treatment with the eGFP, and when we shined the blue light on it, it did indeed fluoresce green. And on the right is this treatment with CBP3 by itself, and they have this very plate-like morphology similar to the type 1 AFP. And then for CBP6, it yielded these larger connected sheets that are hexagonal, and you can see, again, with the eGFP tag, it does fluoresce green, meaning the protein bound, and it looks the same without the eGFP. Right. So in conclusion, we do have these clathrate-dwelling bacteria proteins that bind to and change the morphology of at least this structure 2 type of clathrate. So in the next part, we tested the same CBP, or actually this is an ongoing experiment. I'm doing these in lab now. We're testing the CBPs on methane clathrate, which is a structure 1 clathrate, and this requires a pressure cell, which was very intimidating at first, but we're good now. We're comfortable with it. So this is a stainless steel pressure cell where I basically inject a water droplet on a stage, and you can see it through a sapphire window, and it even has a light source coming in so that we can do a time-lapse imagery with good lighting. And then we pressurize that droplet, either containing the protein or not, with methane gas. And while this is happening, we're monitoring temperature and pressure. So we want to know how these bacterial CBPs affect the morphology of this methane hydrate shell, or methane clathrate shell, they're synonymous, and how they affect the temperature, pressure, stability boundaries. So we monitored pressure and temperature during the entire experiment, but this is during depressurization. So this graph is what you call a clathrate stability diagram. And so we have pressure on the y-axis and temperature on the x-axis. And that dashed line there is the stability line, and above which we have methane clathrate that is stable, and below which it is unstable. Methane clathrate, and below water and gas. Okay. And so this is data of just a DI water droplet that we pressurize to have a clathrate shell. That's what it looks like. It has a regular opaque clathrate shell, and you can see the pressure and temperature data as we depressurize, going from high to low. There's actually that peak there along the stability, or trough, whatever you want to call it, and that is caused when the clathrate is melting, it's actually absorbing heat, and the surrounding is basically getting a little bit colder. And so that star there indicates where we visually see the clathrate start to melt. And this is data for the type 1 AFP. And the morphology is very much different. That droplet, the clathrate shell, has grown these ridges along the side, and it even has like this weird feathery thing that emerged from the droplet. I have no idea what's going on there. We're looking into it. And then where it actually started melting was a tiny bit higher than the stability line, and the overall melting peak was much more shallow, indicating maybe it was a thinner shell. We're not quite sure yet. And this is the eGFP CBP3 data. The morphology is also different than the DI water droplet shell. And where it started to melt, go ahead, was way above the stability line. So the CBP3 both alters morphology and raises the melting pressure. So in conclusion, CBPs also bind to and change the morphology of methane clathrate and also alter the clathrate stability. So implications, of course, are astrobiological because these methane clathrates are extreme environments that are predicted to be on other planetary bodies, and also including Pluto, and then as well as gas hydrate stability. So gas clathrates store gigatons of methane, which is a potent greenhouse gas. As climate warms, that gas clathrate could destabilize, releasing even more methane, thereby accelerating climate change. And of course, these natural gas pipelines, maybe we could use these CBPs and throw them in the pipelines to prevent clathrate clogging. We'll see. We'll try and patent that. So I'd like to acknowledge the NASA Exobiology for funding this research and the GLASS Lab. Thank you so much for your time.